Welcome to the session on bioeconomy and biosecurity. My name is Gene Robinson. I'm the director of the Carl R. Woese Institute for Genomic Biology at the University of Illinois at Urbana-Champaign, and it's my pleasure to be the organizer of this session. A nation's bioeconomy encompasses diverse science and technology-driven economic activity with recent expansions in many areas. We all rely on our bioeconomy to provide our food, underpin our healthcare, produce energy, and many other important materials to safeguard our environment. In all of these sectors, advances in science and technology promise to lead to significant improvements, new medicines, a more sustainable agricultural infrastructure for food and fuel, exciting new materials, and protect our environment. The Earth Biogenome Project, known as the EBP, relates directly to the future of advances in the global bioeconomy. With whole genome sequences for only 0.2% of the planet's eukaryotic species, we've hardly begun to read the books of life that are necessary to achieve the promise of a modern bioeconomy. New insights into gene regulation, genes encoding new products, and new understandings of the genomic basis of adaptation and resiliency are just a few of the many advances that will be spawned by achieving the goals of the EBP. Today's session on the bioeconomy explores a few facets of this rich topic: biofuels, synthetic biology, and biosecurity. For biofuels, there is strong optimism that creative use of plant species, in some cases newly considered for biofuel, will provide basis for ecologically sound and productive new energy sources. Key to this, of course, will be able to have, understand, and carefully manipulate genomes to ensure productive and sustainable biofuel production. Synthetic biology is an illustrious and still new descendant of genomic biology, a creative blend of genomic biology and engineering. Synthetic biology shows how to exploit genomes and promises to make new and creative uses of additional basic genomic information. Of course, genomics and synthetic biology can also be used for bad purposes, can lead to security and economic vulnerabilities around the world. Virulent pathogens can threaten human health or our nation's food supply. Even well-meaning applications of technology can cause unintended ecological problems. And abuses of private genomic information can lead to vulnerabilities in genomic medicine and law enforcement. Our speakers today are prominent scholars and scientists representing each of these three areas and will provide illuminating perspectives on how the bioeconomy is shaping up and what are some promises and perils. We'll start with the perils, biosecurity. Our first speaker is Jacob Sherko, professor at the College of Law, University of Illinois at Urbana-Champaign. He holds a law degree from the University of Michigan Law School and a master's in biotechnology from Columbia University. Professor Sherko also is co-chair of the EBP Ethical, Legal, and Social Implications Committee, dealing with some of the most complex and vexing problems we need to work through to ensure the success of the EBP. The title of Jake's talk is Biosecurity, Laws, Limits, and Best Practices.
Hi, I'm Jake Sherko. I'm a professor of law and an affiliate at the Institute for Genomic Biology at the University of Illinois. And I'm going to be speaking about biosecurity, the law pertaining to biosecurity, limits of those laws, and some best practices going forward. So let's start off with the definition. What is biosecurity? Well, the truth is that even though it's widely distributed, the term is, throughout the literature, there is no set agreed upon definition of biosecurity. It's pretty loosely defined. This is somewhat surprising given that the word seems to be more than 45 years old. It predates even Asilomar. But I think that the clearest, most concise definition of biosecurity probably comes from this WHO FAO report in 2010. It defines biosecurity this way. Biosecurity is analyzing and managing relevant risks to human and plant life and health and associated risks for the environment from biological agents, as opposed to from human agents. What is biosecurity? Well, I think this list of possible issues in biosecurity shows you just how diverse a field it is, just how many different things that quote unquote biosecurity tends to concern itself with. There's a transmission of infectious agents. The COVID-19 pandemic is a perfect example of that. There's biological terrorism. We could think back to the 2001 distribution of anthrax spores to the US Postal Service. This too is a biosecurity issue. We can also think of the accidental release of experimental organisms, some of the concerns raised about the potential release of the Oxitec mosquito or animals that have otherwise been genetically modified for gene drive. Those also tend to get folded into discussions about biosecurity. There's also the risk of lax containment for biological byproducts. That is, if you have a genetically modified organism, it's not just issues concerning the release of the organism itself, but some of the byproducts that the organism may otherwise make, and whether these have the potential to cause an environmental or public health harm. A little bit further afield, right, there are some harms that come from the potential of dual use research. If we are using a biological agent for one particular use, are there issues if we then shift that use to something else? And in addition, even further afield from that, some people would suggest that a lack of adequate access and benefit sharing, as is required under some international agreements, like the Nagoya Protocol, for example, is itself a biosecurity concern. This is a really diverse list of things that quote unquote biosecurity tends to worry itself with. What does the law have to say about it? Well, the laws that we have on biosecurity, they tend to be rather limited in scope. And so here's some cases, or at least here's some elements about exactly how limited that scope is. They tend to be mostly national, right? That is, the laws tend to begin and end at one country's shores. There are some that are based in treaties, that there are some international agreements out there, but even those two, right, tend to be a little bit watered down, as I'll talk about in a moment. The laws also tend to be sectoral. That is, they focus on one particular sector of the law. There is a biosecurity issue in the public health sphere. There's a separate one when it comes to environmental concerns. There's a separate one when it comes to research concerns, right? As opposed to a cross-disciplinary biosecurity law. And much of the laws tend to be post hoc. They concern themselves with what happens after there has been a violation of biosecurity principles rather than ways of preventing those from happening in the first instance. Well, if these are the way that the law governs biosecurity, there's a number of issues concerning that, right? When we're dealing with laws that are national or treaty-based, the reality is that international coordination tends to be extraordinarily difficult, kind of regardless of whatever legal sphere you happen to be in. And even when there is domestic legislation that is implemented as a result of a treaty, the domestic legislation tends to be weak. That is, it tends to not compel its own citizens not to do something as the result of a treaty that they would otherwise want to do. Even if there is a strong law on the books, enforcing that law requires political will. You're asking some politician to enforce the law against its own citizenry. That tends to be tough unless there's a pressing concern. And to be clear, there is no supranational enforcement agency, right? There is no, for example, UN police force that is going around stopping people from engaging in biosecurity violations or preventing those from arising in the first instance. We do have international governance authorities. The World Health Organization is a particularly commendable example of that. But again, this is more in the governance and the suggestion and regulation approach rather than it necessarily is enforcement. If the law tends to be sectoral, this means that the expertise that we may have to combat an otherwise multi-sectoral biosecurity issue tends to be siloed. And in addition, if there's any interagency coordination that is required, that is within one country, if you need to get two agencies to cooperate to police a biosecurity problem, this is going to require interagency coordination. Just like international coordination, that tends to be tough too. And it also very well may raise the issue that if you have two sets of regulations governing the same conduct, that those two things are going to come in conflict with one another. If the law tends to be post hoc when we're dealing with biosecurity issues, this means that it's reactive, not proactive, right? There's little prevention that is actually being done. There's little cleanup that it actually is concerned itself with. There's no reparative mechanism. It's simply punishing the offenders who have otherwise engaged in the harmful conduct. And it's rarely a learning system. We rarely go back and we amend the statute every time we have a particular problem. Like I said before, some of these regulations, even if they do exist and they're enforced, and even if there is cooperation, they tend to be weak or they tend to be underfunded. So usually in problems like these, we're having some kind of international multi-sectoral legal governance issue. Where do we turn to? Generally speaking, researchers turn to private governance. And these are things that we have seen success for in many other areas of genomics and other genomic-related research, rather than necessarily relying on things like domestic legislation or treaties. We've seen this in the context of artificial DNA when we were dealing with some of the issues concerning synthetic biology and the risk of pathogenic release of certain sequences that are ordered. These are the IGSC, IASB principles. When it comes to creating genomic technologies, we've needed many different patent licenses from many different operators in many different parts of the globe. We've had patent licensing entities, right? Essentially warehouses or clearinghouses. MPEG-LA is maybe a great example of this, at least in the internet and video context, right? And when it comes to the sharing of genomic sequences, right, these are all of the country or city principles groups that you are probably familiar with, right? The Bermuda principles, the Toronto principles, the Amsterdam principles, right? All of these are attempts at private governance in this context when it comes to the sharing of genomic sequence data. So what should biodiversity genome sequencers do? I think this can come down to essentially four things, right? Govern, guide, guard, and grow. Biodiversity genome sequencers should govern. That is, we should develop clear and consistent principles for exactly what are the issues one is concerned with when it comes to biosecurity. And these should be published in the primary literature as opposed to being made only exclusive to the particular group or funders that happens to be using them. We should guide other researchers in this particular quest, right? We should be proactive in essentially predicting what some of the problems are going to be in the future. Should really only be reactive if there's an unforeseeable problem that happened. Being proactive means reporting and instituting governance principles on future developments before they actually happen. I think biodiversity genome sequencers should also guard. That is, there needs to be an enforcement mechanism against violators, right? And so that means, for example, not collaborating with institutional, sovereign, or individual researchers who tend to give short shrift to some of the biosecurity principles. And this also means reporting violations, especially to journals. As long as journal publishing is the coin of the realm and editors are gatekeepers to those journals, reporting violations of biosecurity principles to journals is a good bulwark against violations. In addition, the governance should also grow. It shouldn't just be limited to elite members in the field, but by tying together researchers from around the globe who are doing many different types of biodiversity genome sequencing research, we have a better chance of actually effectuating some of this governance going forward. That's it, and I'm more than happy to take questions from anyone who may have them.
Thank you very much, Jake, for that great talk. We'll be holding questions for all of our speakers until the end of the session. Next, it's my pleasure to introduce our speaker related to biofuels. Our speaker is Professor Maureen McCann. Professor McCann is professor and director of the Energy Center in the Discovery Park at Purdue University. She was a member of the distinguished committee that produced a report for the U.S. National Academies of Science, Engineering, and Medicine entitled Safeguarding the Bioeconomy. The title of Dr. McCann's talk is The Biomass-Based Economy. Maureen?
So we have had a traditional bioeconomy for millennia, ever since we first burnt wood for fuel. But a new emerging bioeconomy is enabled by recombinant DNA and by our ability to manipulate genes and genomes. And it's heartening to think that even a decade ago, we didn't have capabilities available to us that are the result of miniaturization and multiplexing of DNA sequencing, the ability to write large synthetic pieces of DNA, genome-wide association studies that allow us to identify at gene resolution the best alleles for a particular trait in a population. We didn't have instrumentation that allows us to go into the field and sequence an organism on the spot, its full genome on a device the size of a flash drive. And we didn't have CRISPR-Cas9 gene editing to allow the facilitation of making engineered organisms. And so technologies of biotech, of engineering, of computing and information sciences are driving a new economic engine. And in turn, those technologies are also accelerating discovery in the life sciences. But this new economy is fueled by sugar instead of fossil fuels. So as we shift from oil, we will shift to measuring our economy and powering it with calories. A recent report from McKinsey Global Institute estimates, from looking at just 400 use cases, 400 potential products from the engine of the bioeconomy, that value of $2 to $4 trillion. This recent report from McKinsey Global Institute estimates, using a study that considered just 400 use cases, 400 possible products from the bioeconomy, that between $2 and $4 trillion of value could be added to the global economy over the next 10 to 20 years. So think about meatless meat, green chemicals and materials, jet fuel produced by bacteria. What's most interesting in this study is their prediction that 60% of physical inputs could be produced biologically. So many of those inputs are currently sourced from oil. From oil, we derive the petrochemicals that give rise to thousands of different chemicals and materials that are the stuff of modern life. And of course, from stuff, we make things. We make carpets and adhesives and rubber and my iPhone. And so as we shift to a bioeconomy and away from a fossil fuel driven economy, one of the largest new resources is sugar. And the largest reservoir of sugar available to us comes from plant biomass. Every plant cell lives inside a box. There are about 40 different plant cell types that give rise to the plant biomass that you see in the world around you. So sugars are built by the activity of photosynthesis in living plants. And I'm showing here 5 of the 40 or so different cell types that make up plants. These are smart buildings. They are built from some of the most complicated molecules in nature. And so it's not surprising that 10% of the genomes of plants are devoted to cell wall related genes, either to genes that are involved in the biosynthesis of those molecules or modifying them for cell growth and differentiation. And the naked plant cell, the protoplast, is not capable of dividing or growing or becoming specialized for its function within the plant without regenerating a cell wall around it. So these smart buildings are highly specialized, they're highly dynamic and responsive. And this then gives rise to a trade-off that we need to think about in terms of how we produce the maximum amount of fuel for the bioeconomy. As we seek to increase the utility of plant cell walls as a raw material for chemicals, for fuels, for polymers and materials, we have to do so without compromising the ability of plants to grow and achieve their full stature. I'm showing here a transmission electron micrograph of the architecture of a plant cell wall. And you can see that it isn't just cement or jelly, it's highly structured. And each of those polymers has a role in being able to allow the plant to achieve its final stature and form in the field. So we have some choices about how to optimize plants for the bioeconomy. We can think about the exploitation of natural genetic diversity in large populations and across pangenomes. We can think about how to optimize cell walls themselves, both for their ability to grow under biotic and abiotic stresses, and also to be optimized for their deconstruction to their component sugars. And we can also think about making unnatural products in plants with the toolkit of eukaryotic synthetic biology. And it's worth remembering in this time of COVID that the anti-Ebola antibodies that comprised the ZMapp vaccine were synthesized in tobacco plants as the production platform. So in order to meet the needs of a growing population, a human population that will be at 9.8 billion by the middle of this century, the biomass that we produce, the potential bioenergy crops, including sorghum and miscanthus and wheat straws, rice straws, fast-growing trees, have to be high yielding. So the maximum number of plants per acre. They have to be cost effective for growers to grow and make a profit. And so we need to be able to diversify the product portfolio that is made in plants. And we need to be extremely efficient about capturing the carbon that the plant has trapped in its body by farming carbon dioxide at 400 parts per million from the atmosphere and converting it into the sugars of biomass. So no carbon left behind, every carbon that is derived from biomass into a target molecule or product. As part of a consensus study run by the US National Academy of Sciences called Safeguarding the Bioeconomy, this study identified a number of risks to the bioeconomy in both failure to promote it and in failure to protect it. And although I've listed all of them here, I just want to say a few words about the three that are highlighted in blue. So full development of the bioeconomy will be impaired if its products and services fail to win public trust and confidence. And so the public trust concerns are going to be numerous. They'll include topics like risks and beneficiaries, the consequences of the release of genetically engineered organisms into the environment, and ethical concerns. The bioeconomy will need to be treated as a component of critical infrastructure, as the electric grid is, as the road systems are, because the bioeconomy will become an ever more significant part of what is now a fossil-fueled economy. And then finally, because this economy is powered by living organisms, fueled by calories, then the impact of climate change on our ability to grow traditional and engineered crops is probably the biggest risk of all. This is the Sagrada Familia church in Barcelona. The masons who built its foundations, its architect, Antoni Gaudi, never saw the realization of a project that takes over a century. Can we use genome science to redesign biomass for food, feed, fuel, pharma, and fiber? Can we do it while we increase yields to meet the needs of 9.8 billion humans? And can we do a better job of communicating our science to the public and to decision makers. As we redesign the genomes of living organisms to change the agricultural landscape, the energy system, and to build a new global bioeconomy, it's on all of us to make sure that the foundations are strong.
Thank you, Maureen, for that great talk. To remind everyone, we'll hold questions until the end. It's my pleasure now to introduce our final speaker, who will be talking about synthetic biology. Dr. Patrick Boyle is one of the leaders at Ginkgo Bioworks Incorporated, one of the pioneering and leading companies in the burgeoning synthetic biology sector. His training at Harvard and MIT included a postdoctoral stint at the Weiss Institute for Biologically Inspired Engineering, one of the real hotbeds in the world for synthetic biology innovation. A few of his roles at Ginkgo are head of code base and organism designer. So you can see it's not business as usual at Ginkgo. Patrick also was a member of the Safeguarding the Bioeconomy Committee. He will speak about some of Ginkgo's efforts to grow the bioeconomy with new genomic-based applications. And if you'll just bear with us for a minute while we switch platforms, the talk will be appearing live momentarily.
Hi, everyone. I'm Patrick Boyle, and happy to talk to you about Ginkgo and our role in what we feel is the growing bioeconomy. I think to understand Ginkgo, you first have to understand Tom Knight. He's one of our founders here. Tom actually spent 30 years in electrical engineering at MIT before switching into biology. So he taught the semiconductor design course at MIT, really cut his teeth in the 60s, 70s, and 80s developing computer technology. And the reason that he became interested in biology is that he realized that biology is really the only technology that exists that can place atoms with the precision that we need to get to the end of Moore's law. So if you think about transistors getting smaller and smaller, the only way to do chemistry at the length scale of atoms is really with biology. So in the mid-90s, he actually retrained himself as a biologist. He set up a wet lab in the computer science department at MIT. Was really responsible for founding the field of synthetic biology, which fuses engineering principles with genetics, genomics, and all the tools that we have today for engineering biology. And this philosophy really permeates our approach in terms of how we engineer biology at Ginkgo. This is just a quick snapshot of what Ginkgo looks like today. So the company was started in 2008, have been growing a lot since then. In fact, the slide is out of date. It should say 500 people now. But Ginkgo is based in Boston. We're, as you can see, very focused on automation. So Ginkgo accelerates the engineering of biology by applying both software and hardware automation to speed up and scale up the process of engineering biology. So to give you a sense of scale, when I did my PhD about 10 years ago, I designed and synthesized 6 genes as part of my PhD and spent most of my time pipetting clear liquids around by hand to build DNA. Today at Ginkgo, in a typical month, we're synthesizing anywhere between 5 and 30,000 genes per month. So, again, the applications of automation has really changed the way that we can engineer biology. We are, in fact, the first biotech company in an accelerator in Silicon Valley called Y Combinator. You probably know them more for helping start companies like Airbnb and Box and big tech companies. But this is really indicative of the fact that the tech community and investment community is waking up to the applications and power of biology. And in fact, Ginkgo has been able to raise more than $900 million in private investment over the last 5 years. So why should we care about the bioeconomy and why should we want to protect it? Well, our whole thesis is that, the bioeconomy of today is a small fraction of what the bioeconomy could be in the future. So if you think about synthetic biology as revolutionizing and making more sectors of the economy accessible to biology, we feel that that economy should grow considerably. So, you have the traditional sectors of pharma, agriculture, and industrial biotech, where biology is prominent today. We feel that, manufacturing, textiles, consumer electronics, these are all areas that could be accessed by biology and are being changed by biology because fundamentally biology is a technology to manufacture stuff. Why today versus 10 years ago or even 20 years ago? I think one of the things that's been really exciting in the field is that we're finally at the confluence of rapidly improving technologies that, when put together, allow us to do things that weren't possible 10 years ago. So, folks in the genomics community here are probably very familiar with the curve on the right, showing the dramatic decrease in the cost of DNA sequencing. For us as programmers, we think of programming cells as, can you read and write code, aka can you sequence DNA and can you synthesize DNA? Perhaps a slightly less well understood curve is the cost of DNA synthesis. So Ginkgo actually is the world's largest consumer of DNA synthesis. We estimate probably about a quarter of the world's DNA synthesis comes through Ginkgo. And these two technologies together, which are improving faster than Moore's law, means that year over year, our ability to engineer biology is improving at a similar rate. So the chart on the left is a chart of output from our facilities, which we call foundries. And what you can see is that the number of laboratory operations that we're able to do each month is increasing at a rate similar to Moore's law. So from our perspective, this means that year over year, we can do much more work, which means we can work with more partners and get more projects done. So just an example of some of these partners to illustrate how the bioeconomy is changing. One of our partners in San Diego is a company called Genomatica. They engineer bacteria to produce novel materials. So for example, a lot of replacements for things like plastics or even stretchable materials like spandex, they're really revolutionizing how we can produce those materials sustainably using fermentation. Synlogic is a pharma company opening up a new modality for pharma. So they actually work on engineered bacteria as living medicines. So for example, we're working with them to engineer a whole host of microbes for novel applications. One common area for them is metabolic disease. So if you're familiar with a disease called phenylketonuria, this is the inability to metabolize phenylalanine, which has devastating effects for the people with this genetic disorder. The approach that Synlogic uses is introducing engineered E. coli into the gut where E. coli lives to replace that missing metabolic activity. And what's exciting about this is that there's a whole host of other metabolic disorders and other diseases that can be addressed by a similar way by engineering bacteria that live in your gut. And this is work that is in phase 2 clinical trials right now. Also in traditional sectors like agriculture, there are places where synthetic biology can have transformative applications. We have a program, a joint venture with Bayer Crop Sciences, for example, to engineer microbes to fix nitrogen from the atmosphere for non-legume plants. So plants like corn, wheat, and rice that require a lot of fertilizer. And of course, this group likely knows that fertilizer production generates about 3% of the world's greenhouse gas output. A lot of new areas for biotechnology also opening up. We have a partnership with Kronos, which is one of the largest cannabis companies in the world, to engineer yeast to produce cannabis. So in addition to THC, there's a whole host of rare cannabinoids that are inefficient to extract from the current plant. So effectively, the industry is moving to produce these cannabinoid products via fermentation rather than via plants. And then, one more sector to go over that's novel over the last 10 years. Ginkgo has spun off a company called Motif Foodworks that works on developing novel plant-based foods. So if you're familiar with things like the Impossible Burger or Beyond Meat, these are all products that mimic meat but have no meat-based products in them. With Motif, we're partnering to engineer microbes to produce a lot of the key proteins and other ingredients that are needed for really satisfying plant-based foods. So one important topic that's hard to escape right now is COVID-19. I think the other reason why we're very excited about this growing synthetic biology sector of the bioeconomy is that it really provides a flexible capacity to apply to big problems in the world. So for example, when COVID-19 hit earlier this year, we actually paused many of our commercial programs in order to free up capacity in our platform to respond to the COVID-19 pandemic. In fact, we dedicated more than $25 million of our own funding to partner rapidly with companies in this space so that we could accelerate the development of products that could help turn the tide of the pandemic. Just a couple of examples of where we're working there. One is in the therapeutic space, namely therapeutic antibodies. So Berkeley Lights is another exciting company in our sector that's really revolutionizing optical microfluidics for rapid sorting of cells. This is great for identifying new antibodies, and we're partnered with them and a company called Totient to help them develop better antibody therapeutics. We're also working to improve the vaccine manufacturing response. So back in April, we partnered with Moderna to help them improve the manufacturing process for the raw materials for their vaccines. Again, this is a technical area that is very close to Ginkgo's core competency, and we were able to jump in very quickly and help them optimize those processes. And since then, have actually expanded this work to support other companies in this space as well. And then our major project in this space is accelerating the development of diagnostics. So we've actually launched a new brand called Concentric by Ginkgo Bioworks. This is a service that helps back to school and back to work-based testing for workplaces. And it's really centered around next generation sequencing as the core technology. One, because that's an area that we've applied a lot of automation to just as part of our normal business. And two, because we think it helps reduce the supply chain constraints on traditional diagnostic testing. So again, we're in the midst of building a facility to support hundreds of thousands of tests per day. As a highly automated facility that will take about 100,000 square feet in our facility. And in fact, we're very fortunate to be selected by the NIH as one of their RADx recipients in order to accelerate the development of this technology and deploy it by the end of the year. So, importantly, I think for this topic, we want to talk about biosecurity in the context of the bioeconomy. For us, this is something that's not optional to develop as core platform developers in this space. Is really analogous to developing computer technology in the 1960s or 70s. It's really important that we try to build good security practices into this economy as it grows. Just a couple of quick examples here before I wrap up. In addition to the bioeconomy NAS report that Maureen talked about, there's an earlier report that I contributed to around biodefense in the age of synthetic biology. One of the challenges for people who are concerned about this space is how to keep up with the rapid change of technology in the space. So what this report developed is a framework that allows analysts to piece together the different aspects of new technologies in the synthetic biology space and help build out updated risk assessments for new technologies. So for example, how does an advancement in CRISPR change your ability to engineer bacteria? The framework developed in this report is meant to help people have a living document that keeps up with those changes. And then finally, just briefly on the Safeguarding the Bioeconomy report, Maureen already spoke about how this report, talks about potential areas of concern. I think from the Ginkgo perspective, I think, we use computation and software as a big piece of the way that we're able to do the work that we do. So that introduces new threat vectors. So in addition to being concerned about applications of engineered biology, we should also be concerned about infrastructure and how that impacts the bioeconomy. So for example, cyber risks is something that's very important for us to be concerned about. So with that, I'll stop there and looking forward to seeing all of your questions. Thanks.
Great. Thank you very much for that great talk. We will now take some questions. So here's a question for Maureen. Maureen, what are your thoughts on algae-based biomass economy?
So I think it's important that we develop numerous different platforms because the risks of an environmental impact, for example, on a field of switchgrass or of any other bioenergy crop, could mean an interruption in the supply of those sugars that I talked about as being very fundamental for any sort of fermentative production process or a chemical catalytic process. And so having numerous different platforms, including algae, is going to be key to resiliency, being able to shift the production of a particular product from one platform to another so that supply is not interrupted. Algae are terrific. They have their own risks associated with them, for example, the crashing of open ponds. But I think that the tools of synthetic biology and the improvement of these organisms are helping to mitigate those risks. So I'm a fan of as many different production platforms as possible to improve the resiliency and the flexibility of response.
Great. Thank you. A question for Jake. You talked about barriers at the international level in terms of the uniformity of laws. I was wondering whether you could compare and contrast the IP landscape, legal landscape, with respect to cooperation across the world with the landscape for personal genomic security information.
Sure. So the landscape for IP harmonization is somewhat of a mixed bag, at least with respect to some of the sequencing that many people in this group are probably interested in. So there are harmonized intellectual property laws across, almost 130 different countries now, and that makes it a lot easier to file for a patent application in one country and have it have effect in many other countries around the world. The one exception to this, perhaps somewhat ironically, is when it comes to inventions that pertain to genomic sequencing. They have, for the most part, been tamped down here in the United States. They are similarly restricted elsewhere in the world, but they remain available in Europe. So this greatly complicates the IP landscape when it comes to patents and protecting things that pertain to genomic sequencing. With respect to doing that, when it comes to some of the personal genomic data security issues, we have never had an attempt to harmonize a lot of these laws across the world. But the thing that truly seems to be happening now is that the GDPR really seems to be the law that is, forcibly harmonizing some of these efforts everywhere. So I think the place to look at when it comes to harmonization and when it comes to personal genomic data security are probably going to be implications with respect to the GDPR, whether the sequencing or whether the data is stored in Europe or elsewhere.
Thank you. A question for Patrick. Obviously, ginkgo depends on nature for raw materials that it then ingeniously goes about engineering. I was wondering, this is my own question, does ginkgo have its own bioprospecting division or program to look to novelty in nature, species with some new wrinkles that haven't yet been exploited yet? Or is it, how do you go about bioprospecting?
Yeah, that's a great question. And, initially we had actually depended, largely on public, sequencing data sets, but obviously that's only a small fraction of what's out there. So over time, we've invested more and more in that type of sequencing. In fact, one of the core reasons that we're developing NGS-based diagnostics is the fact that we already have a very large-scale automated platform powered by the Illumina NovaSeq that's sequencing thousands of samples per day under normal operations. So that's been an enormously useful way for us to find new content. Obviously, there are concerns, such as Nagoya, to make sure that we're appropriately respecting and providing benefit sharing to source countries. So we take that into account for any sort of bioprospecting work that we need to do. Is making sure that we're dealing with the respective local authorities before we do any of that sort of work. One quick example of that is that we acquired a company called the Warp Drive Bio about a year ago as part of our work in antibiotics development. We're using their genomic databases to develop possible new antibiotics in partnership with Roche. That acquisition brought in more than 100,000 unique genome sequences that basically they had built as a genome mining platform to look for novel antibiotics. So the useful thing for ginkgo is that all of these data sets are broadly useful. So it's a growing area of interest for us.
Mm-hmm. And a related question from our fearless leader, Harris Lewin. You just mentioned the Warp acquisition. In addition to that, are there key groups of organisms that you would like to see sequenced for current synthetic biology efforts?
Yeah, I mean, I'm pretty ambitious here, so I think maybe we could just start domestically and go from there. But I don't see any reason why we shouldn't be at least trying to sequence soil from all 50 states to begin with, and go from there. I think one of the things that we've realized is because of DNA synthesis, the ability to write DNA, we can really try out lots and lots of different designs inspired by nature. And the more diversity we have to enter into those processes, the better. And things like NCBI GenBank, they're very great databases, but they basically represent the interests of funding agencies, which makes sense. So they're overfit to, for example, medical or biofuels type sequencing. So I think there is an opportunity to just broadly say, let's try to sequence everything because it's generally useful to the bioeconomy, and it's also very important work to do ecologically as well.
Thank you. Here's a question for you, Jake, from Anonymous. Do you think some political claims may be also considered to be regulated by biosecurity policies?
Some political claims be regulated by biosecurity policies. That is, I just want to make sure that I'm understanding the question, right? That claims to the propriety of a certain political group happen to be regulated by some biosecurity policies. I mean, sure. Certainly, although, I mean, I think relatively around the world, it's rare, right? I mean, we've seen some kind of major upheaval in Bolivia in the past few years, where I think some of the populist movement is under the impression that there's biopiracy going on with respect to its country. And so by virtue of that, we're seeing some political parties attach themselves, to that particular claim. We've seen this to dramatically less effect in New Zealand, for example, where I think these issues tend to arise, but then somewhat peter out. They don't necessarily make majoritarian policy. So, I mean, I think it's fair to say that it probably happens. It's probably a question that's best suited, frankly, for a political scientist, and that person is not me. But I mean, they do seem to occur, even if they are not frequent.
Thank you. And Patrick, you mentioned the Genome Right project. I was wondering if you could speak to that for a moment. Is Ginkgo involved in this? This is a public project, grand challenge level to learn how to write DNA at really diminish the costs associated with that. Is Ginkgo involved with that? And do you see some parallels between that project and the Earth Biogenome Project?
Yeah, so we're not officially involved in the Human Genome Right project. We're paying a lot of attention to it. It's a very, very interesting project. And I think in general, where the field is moving is towards the ability to basically write chromosomes, particularly microbial chromosomes, at will. Typical synthesis for us is hundreds of sequences up to 10 kb in length. So you can get to chromosome style synthesis over time that way. But I think in general, the transition, I think, is really towards what is your genome of interest, and how do you write it? I think the era of making edits on genomes won't last for very long, just because it's getting much easier to write. But sorry, I'm not sure if that answered your core question, though.
Yeah, that's fine. Let's see. We've got one more minute, and we do have one more question. And I guess it's appropriate that it's related to COVID-19, and that relates to shark material for vaccine development. Patrick, do you see a role for that?
Yeah, so this is an interesting one. So GSK and others use shark liver oil, a product of shark liver oil called squalene. That seems to be a pretty good adjuvant for vaccine development. Obviously, you don't want your bioeconomy to depend on scaling something by killing more sharks. The good news is that there are a number of companies that have been able to develop routes to producing squalene via fermentation. So from my point of view, that's very important, very attractive way to scale the production of key adjuvants is via fermentation. One, because you don't have to kill sharks, but two, it's also just a much more scalable supply that you can turn on and off on demand.
Great, thank you. Well, we've come to the end of our time. I'd like to thank our speakers, Maureen, Jake, and Patrick, for really great talks. And thank you, the audience, for your questions and interest. And take care. Stay healthy. Bye-bye.